ago. So she will present her research on ecologizing and humanizing biodiversity database, citizen science and environmental stakeholders in Taiwan. So now your time, Serena. You can start, share your screen and unmute yourself and start your presentation. Thank you.
All right. Thank you and hello.
Yeah.
Can you see my
I'm sorry. I'm sorry to all participants, please mute yourself during the presentation.
All right. Can you hear me and see my screen?
Yeah, I hear you now. Yeah, your screen good.
All right, wonderful.
Yeah.
All right. Hi, everybody. My name is Serena Calcagno, and I wanted to start out in the spirit of the East-West Center and this conference today with a brief land acknowledgement. Because we are remote, I'm joining you from Santa Cruz, California, which is where I grew up, and also where I live right now. This land is the historical land of the Owasso people, also the Awotos. They're sub-tribes of what you may know as Ohlone. And I just wanted to begin with that, since today we will be talking about things that relate to traditional peoples and land sovereignty. But I also wanted to throw it back to last year when I went to the IGSC for the first time. It was truly such a pleasure, and I wanted to thank you, moderators and organizers and everyone at the East-West Center, for making this possible despite the distance, despite all the tech challenges, and also acknowledge how formative this conference has been in my own studies. When I was just beginning my research, which was basically kind of part of exploring the stories of a town that I lived in for a year while I did a Fulbright English teaching grant, it really gave me a powerful chance to start getting other perspectives on what it takes to do a good interview and to really learn from a community. So that's what I'm hoping to do today, learn a little bit more from all of you about how to improve even my relationship with the community in a research capacity. So brief roadmap of what we'll do today. You know a little bit now about my positionality, but I'll share a bit more about that year in Taiwan and how it brought me to study what I do today. I'll also talk about the project that I applied to this conference with, the sort of ecologizing and humanizing biodiversity-based databases project, as it connects to the above positionality and sovereignty piece. And then I want to talk about the next steps in my research, and I really just want to give you guys an idea of where I'm trying to go next, and gather some feedback from you about how to go forward in this project. But first, I wanted to introduce my most recent piece that emerged from the conference last year. You can see the title up there. I won't read it out loud. But I wanted to begin by acknowledging that when I was in Taiwan, I learned really that citizen scientists, people who go out and count what they see in nature and upload it to database, a lot of these people are environmentalists first, activists first, and people who care about their local spaces first. And so I wrote an article about that emerged from that paper, from that presentation, and from really my relationship with my birdwatching teachers from when I lived in Taiwan. So that's kind of how this all began, to talk about my current project and how it emerged from that interview project that really did a critical kind of look at the power structures involved in science in Taiwan and broadly in Asia. It's really emerging from citizen science, which is a new turn in the way science is done, especially gathering information about biodiversity. But maybe you're not familiar with this term, citizen science. It sounds maybe a little political, a little bit futuristic, and it's really the entire scientific process, but done by average people. And citizen science is a problematic, contested term. Really, citizen is a confusing one to engage with science, right? But we also can't ignore that nations and states are almost inextricable from the way science is done in most places. And so we're going to look today about how we might take the infrastructures that exist and make them a little bit more grassroots and incorporate the meanings of actual people into what they do. So this is where that comes from. When you're gathering citizen science data, it's a community event. You're going out with people, you're seeing what you can see, you're counting it, pointing it out, identifying it, and verifying it, and then uploading it with not just the identification, but the geotag, and then the date and place, date and time. And so these are some of those people that I did this practice with, and I learned what citizen science was really from engaging with them. And you can maybe see over here, the person who I wrote the article about, she was my birdwatching teacher from the beginning. So that's Huang Shumei. And really I learned from them how to begin this process and start thinking about how data is really about sovereignty and protecting your land, your space. So we can see open data, open science, citizen science as not really, it doesn't have to be about the citizen piece, but that's just an interesting lens to look at it from. And so I wanted to just engage with the idea that indigeneity, local peoples, traditional peoples are invested in this idea. So it connects to Hawaii itself, island nations, all of these kind of concepts by way of giving people open information to use potentially to say, you know, if a government wants to build something where you are, you can use the data, biodiversity data, to justify why they shouldn't build there. So this is kind of the grounding of why citizen science is important, whether or not actual citizens are doing the work. And so I analyzed a database that has emerged in Taiwan using kind of three pillars. And I don't want you to get lost in the detail here, but I do want you to see that there's three main pillars of kind of content that I used. That's the practice of citizen science, the data that's collected through it, and then looking at kind of the relationship between the state and environment. And I used this kind of core here, these four sources, to look at the status, history, and kind of potential future of citizen science and open science generally in Taiwan, and examining the database that has emerged. The kind of critiques I won't focus heavily on in this talk, but if you're interested in my paper, I hope that you can email me and I'd be happy to share. But that's kind of the structure of what I'll share with you about my project. Just a little bit from each of these, and then sort of how it connects to citizen science as it is done in Taiwan today. So first I want to acknowledge the elephant in the room, citizen. So I'll just read this quote. When we talk about citizen science, we cannot get around the concept of citizen. It's a political concept, category, and entity. And when one uses the term, one is willy-nilly making a political statement. One is defining citizenship, potentially drawing political boundaries around it, just as one may be trying to widen participation in science. So science, technology, and society scholars in Taiwan are recognizing this awkward potential of this word. And I think it's interesting as well to see how the ideas of citizenship are also wrapped up in biodiversity. So moving kind of from that to the ways that you can understand citizen science broadly, it's tied up in different entities, right? Like globally, there may be interest in promoting citizen science because it's part of making sure we can face climate change. And who doesn't want that, right? But you know, as a nation, maybe Taiwan wants to use citizen science to put themselves on the map, to get recognition, to have others see the biodiversity that the place is characterized by. And then maybe on a smaller scale, a local community might be concerned with the thing I mentioned earlier about protecting their community from encroaching development. And so these are sort of some of the ideas that are wrapped up in different formations of citizen science and the imagined communities, you know, Benedict Anderson, classic kind of example. You can just, you know, these are just examples, but I think it can get your mind turning about why different ways of doing citizen science are really wrapped up in greater power structures. And how really do we then make sure that citizen science is empowering the people kind of in the neighborhood, making sure that we address science from a grassroots level. Next, we can look at the data side of this. So we, you know, I don't think I'm speaking to anyone who wouldn't agree that science has often been colonial. People who've gone on expeditions to learn about the natural environment in different places and surveying biology are often alienating labor from human bodies and bodies of indigenous people, bodies of Native people. And in that process, we've kind of turned into this historical, we've moved into this historical moment where the data is more important than anything else, more important than the people who are actually there showing you what exists. And so this is a big problem to face, the datafication and overvaluing of data beyond livelihood. But citizen science has really like become the dominant way to gather this information. And so that's to say that community labor is the foundation of this, that citizen science is predominantly the way we gather what is, viewable biodiversity. So of course, like we record what we can see, birds, animals, much easier for a human to see than microbes, which you'll be hearing about in the next talk. But this is a global issue considering how biodiversity is recorded. And like I said, wrapped up in climate change. If we think of biodiversity as sort of an indicator of resilience, places that have the most biodiversity will likely be the most resilient to climate change. It's a problem then that we have all these spots in Africa and in Asia, way up in the north that don't have very much data. And so how do we then empower the grassroots act of this without having it be co-opted by global governments? But take a look here. Within Asia, we see this crazy hotspot in Taiwan, and that stands out, right? Within Asia, both India and Taiwan are kind of at the forefront of digitizing their biodiversity information and making it public. So when we think about that connection between states and biodiversity, the state is sort of serving as this, like a receptacle, a storage facility for this information. And that shows that it's seeing itself in a particular way in relation to the environment. So there's two different kind of possibilities here. I think it's one or both of these. It could be an environmental state, a state that accepts the provisioning of environmental collective goods as a core responsibility. Or a little bit more pessimistically, we could see Taiwan as transitioning from a developmental state simply to an eco-developmental one that is still very concerned with development beyond anything else, but really wants to grow their political legitimacy while they're, using development to continue to have growth. So kind of unsure about whether it's one or both of these, but I think that there's a strong indicator that it's at least one. So to introduce the database, hopefully you'll get that in the chat if you would like to take a look for yourself, but this is where Taiwan is storing all of the citizen science information that's been created by average people, including the stuff that I contributed to with my friends in Meinong. And it's consolidated under the Agricultural Committee of the Executive UN, which is kind of weird, right? Like, why would this be wrapped up in agriculture, right? But really it's because the idea of dealing with climate change is often given to the agricultural committees of the world. And like what they're really doing is just gathering that data so they can give it to scientists. So the role of these biodiversity databases is really all about kind of fueling publications. So that's how the biodiversity database sees itself. But how do we then kind of like go from its accomplishments, which are many, and I won't read these out to you, but other than making Taiwan's biodiversity visible, how does this data give back to average people? And how do we make this site really represent their needs more? So that's an example of how the data is really just a reflection of human activity is here. So if you take a look at these two adjacent squares, this one, it makes it seem like there's only 19 data points, and it was funny to me that there's a shrimp in this inland area, but this is very few observations at all. It looks impressive on the map, but this is really not that much data generally, and the resolution is pretty vague. And just adjacent to it, almost 350 species. So this is not a real reflection of true biodiversity, but it's a nice indicator. And I want to just like call out that this is the product of probably a community that has gathered repeatedly in the same place to show, respect to the land in the place they are. So my limited critique, without rehashing my entire paper really, is that if you take a look at this site, it's relatively shallow ecologically. It doesn't show all that much of really what's going on. And it also obscures the community labor that's behind it. It's a warehouse, not a museum. It's not educational really to any degree. It's more about Taiwan's government saying, here's where you should go to get more data because we don't have data here. And it privileges those national and regional needs over why people are actually doing citizen science in the first place. And finally, like you've just seen, that beautiful data might not be all that representative. It might still have a lot of imperfections. So my next steps, the thing that I would really love to hear your feedback on is sort of where I want to go next in terms of surveying how people in these communities, particularly the community I was in, the Hakka kind of inland minority community that is collecting so much data, what do they really want this site to be? I want to gather suggestions from them about what they think when they see this site, kind of see how they move through the site and understand its about page, especially like, do they want it to be all about publication or do they want it to be about something else? And then I'd like to compare it with some other sites, kind of how they do what they do elsewhere and how this particular site could be improved using that common ground, especially to give respect to the people who collect the data and also use it to do what they want it to do. So finally, just a brief thank you to all of you for being here. Thanks to my professors, Brian Comey-Dempster, Matthew Berry, and Professor Genevieve Leung. And yeah, thank you guys for your help and your feedback today. I'm looking forward to talking more.
Thank you, Serena. Excellent timing. Yeah. So yeah, please save your questions until the end of our presentations. So you guys, if you want to ask a question, you can put the question in the chat box of Zoom or Hoover, or you can raise your hand, and then I will call on you later. So for now, we want to move on to the next presentations. So I'm glad to introduce our second presentation. So now we have Kimberly Sampson with her presentations on the biology and aquaculture potential of an endemic fission fish, Mesopris canary, family Theraponidae. So please welcome Kimberly Sampson from School of Marine Studies, the University of South Pacific. So Kimberly, now are you here, Kimberly?
Hi, sorry, yep.
Oh, yeah, so please share your screen. Yeah, thank you. So your time.
Are you able to see my screen?
Yeah, I see your screen. Can you make it larger?
Yeah, one second.
I put it in a viewing. Yeah, good now. Yeah, and remember to print down the IGSC timer. Yeah. Thank you. So your turn.
Sorry, one second. All right. Hi, Bula, aloha, hello. My name is Kimberly Sampson, and today I will be presenting my research topic, which is the biology and aquaculture potential of endemic Fijian fish, Mesopristis.
Sorry, Kimberly.
Yeah.
I think your slide still in the review.
Oh, sorry.
Yeah, I don't know. We don't see the slide, so but see the whole. Yeah, if you want to let it like that, it's okay.
Okay.
Yeah, Kimberly, now we can see your notes on the slides. Do you want to play that?
Yeah, I'm not sure how to just share a portion of the screen. Sorry. Okay,
I
let me
think,
just.
yeah, I think it's good
perfect.
now. Yeah.
Sorry, one second. I need to.
Yeah, no worries. Just, yeah, take your time and start.
Oh, right. Okay. All right. Is that good? Can you guys see that?
Yeah, good. Here you go.
Yep. Okay. All right. So as I was saying, right, to begin, in the year 2019, the world's human population reached 7.7 billion, which 8.9% or 690 million people were estimated to be undernourished. The global human population is expected to further increase to between 9 and 11 billion by the year 2050. This has resulted in a major global challenge of finding solutions for feeding such a large number of people, alongside combating other factors such as providing the correct nutrition, addressing climate change, overexploitation of the environment, destruction of natural resources, as well as economic uncertainties. Profound changes will need to be made to satisfy the food and nutrition needs of the Earth's growing population, and as a result, the ever increasing demand of nutritious food. These changes will likely involve the type, availability, and how the food is produced. And for these global food and nutritional security, there must be an increased production and quality. Global food production comes mainly from three sources: terrestrial agriculture, livestock farming, and the capture of aquatic animals. It has been widely reported that the capture of aquatic animals has reached the limits of these natural ecosystems that can provide, and many species are either endangered, near extinction, or already extinct. So let's focus our thoughts on the Pacific region, where I come from. Generally, Pacific Island nations have small populations with few terrestrial resources due to the scarcity of land. These factors contribute to the restricted economies of these small island nations. So these small island nations have been left no choice but to use the one important resource that all Pacific Islanders have, which is the ocean. Countries such as Nauru, Tuvalu, and Kiribati have derived a large portion of their gross national product from the sale of tuna fishing rights in their EEZs to distant fishing nations. Ideally, if these fisheries were better managed, um, then the income generation from these resources could be sustainable. However, unsustainable fishing has continued to be practiced over the years. So what is the solution? There are a few solutions such as fisheries management, well-executed restocking programs, and aquaculture. Which brings us to aquaculture. What is aquaculture? It is the husbandry or farming of aquatic plants and animals in all types of water environments for commercial, subsistence, and recreational purposes. Aquaculture has become important an important industry globally, specifically in developing nations, which are using the sector to battle poverty by providing food security and income generation. It can alleviate stock depletion through methods of restocking, stock enhancement, and culture. And um, these will benefit our island nations by increasing income generation, employment opportunities, more importantly, food security, and conserving biodiversity. So there are positive attributes towards aquaculture in the Pacific, such as the high demands and close proximity to major markets. Therefore, it can easily be developed and achieved to its full potential. However, there has been unsuccessful ventures of aquaculture in the Pacific, and this is due mainly to constraints faced. So there is a need to develop the aquaculture sector in the Pacific, and it's crucial for the long-term food security against the threat of climate change, as well as to maintain a traditional diet of seafood, which is no longer easily available. Um, and so for the aquaculture sector to develop and expand, there needs to be a diversity of suitable species for cultivation. Therefore, introducing new species will provide a range of products. So when you're introducing new species, globally, it has been common to introduce a species to a new geographical area. This is frowned upon because non-native species in some cases can cause harm to the environment. Therefore, when considering a new species for aquaculture, it is essential that responsible practices are in order to protect the biodiversity of the country and the region. And in the Pacific, particularly due to the geographical spread of the islands, the capacity for maintaining national biosecurity is limited and in some cases non-existent. So the ideal practice is to focus on a native species rather than an introduced species for aquaculture. So because there's such a high biodiversity, it's no surprise that there are a few species that have potential for aquaculture in the Pacific. One such species is Mesopristis neri, which is an endemic species to Fiji. It's known locally as reve, and previous studies have noted that there are a few characteristics that suggest the potential for aquaculture. So this study investigates habitat conditions, diet preferences, and reproductive seasonality to determine the aquaculture potential. So Mesopristis neri, just a brief background. It's commonly known as the orange spotted terrapone. Locally in Fiji, it's known as reve or ngai waururu. It is one of the two species of the family that are found in Fiji. It's found locally in markets. It's eaten as a fish food, and it has quite an appealing appearance in comparison to most brackish water fish, with deep orange brown markings against the silvery body and yellow fins. So the study location was the Navua River. It is one of the largest watercourses in Fiji, and it's located on the eastern side of Viti Levu, not far from our capital, which is Suva. 3 sites were selected. Site 1 located at the town center, which is the closest to the river mouth, and site 3 being the furthest upstream. The time frame for the project was 12 months, sampling 4 times a month. A total of 80 fish were caught of various sizes. Habitat information was collected at each site during each sampling, and fish were euthanized and dissected for diet and reproductive studies. Now, to begin with distribution, most fish were caught from site 1 over the 1-year period. There was a significant capture between sites found, however, no significant difference between months. This showed us that there may be no seasonal occurrence of reve, but it may have a preference to the particular site, which would be the lower estuarine section of the river. In terms of size, there were no significant differences between size classes and sites, meaning they weren't dependent on specific areas according to their age or size. This study also confirms that this species is a Eurohaline species, meaning it's found in brackish waters. For the diet study, according to an index of relative importance, the top 10 diet categories were a mixture of both vegetation and animal material, and this indicated that the species has an omnivorous diet. In terms of weight, there's a relative importance of crab, which comprised of the highest weight, comprised of the highest weight, and the second, clams. This weight analysis provides an information on the nutritional value of the prey item. So this finding is significant in aquaculture as it assumes that fish are able to ingest foreign food items, indicating a versatile feeding range. Another study had also shown that it is able to ingest an artificial feed pellet, which is also important for aquaculture, as we know that it's able to ingest feed, which is important. So reproduction, there was a total of 80 fish. Of the 80 fish, about 60 were females. So the sex ratio is 1:3. The females in the study were selected and a GSI, or gonadosomatic index, was calculated. This showed us that there was an increase in GSI values in the cooler months and it dropped in the warmer months. What does this show us? That there is possibly a spawning season within the warmer months and gonad maturity within cooler months. So within the family, only a few species have undergone this type of study. And studies that have been conducted have also indicated this spawning period in the warmer months as well. So the main findings of this study, first of all, this is the first study of the species to this extent in Fiji. It's an endemic species again. This study conclusively demonstrated that the species is a Eurohaline species. The diet study indicates that it has an omnivorous diet, also with the possibility of shifting from a carnivorous juvenile to an omnivorous adult. The reproductive study shows that spawning is likely to occur in warmer months and fish maintain maturity in the cooler months. The identification of this spawning period is vital in the production of stock for aquaculture, with temperature possibly playing a huge role in its reproduction. So during the study, there were two major constraints. One was the rate of the fish that were able to be caught, and two was getting fish under a certain size class, which in this study was below 20 centimeters. The study attempts to first, as an endemic species, find if it has potential for aquaculture, apart from its identity and basic information. The information gathered is important for the industry as a whole, and also for just the country's basic knowledge on their biodiversity, because it's an endemic species. So based on these study efforts, a subsistence or small scale commercial level farming operation would be viable in our small communities, village communities. So yeah, I'd just like to thank the Australian Centre for International Agriculture Research and the University of the South Pacific for helping me carry out this project. And that's it. Thank you.
Thank you, Kimberly. Yeah, you still have time, but yeah, thank you for interesting presentation. And again, if you guys have any questions, please put it in the chat, and we will go back to it later. So now, yeah, now we have two presenters with their research on exploring microbial indicators of coastal ecosystem health across human yield gradient on Tutula, American Samoa. And please welcome Alice Hawkins and Becca Lansing, both from marine biologists at UH Manoa. Yeah, I think we will have the pre-recording video to squash now. So I don't need the timer, I guess. Yeah, so Megan, will you play the video? Thank you.
Yes, and can you let me know, just make sure and confirm that we can hear sound on it as well.
Perfect. So let me go ahead and pull it up then. Okay, so whoever is sharing screen right now, I think it's Becca, yeah? Okay, perfect, thank you.
Typically when you share screen, there's an option to share computer audio. Okay, I found it. Let's try that again, sorry.
Oh, okay, I can also do it from my end if you would prefer. Indicators of coastal ecosystem health across a human use gradient on Tutuila, American Samoa. And it should be noted that Nalani is calling in from a field station on the Big Island, so her background is not virtual versus mine very much is. All right, so a commonly known source of nutrients to coastal reefs is through runoff of rivers and streams. However, a less known source of nutrients to the coastal ecosystem is through the infiltration of these nutrients into the groundwater and then the discharge of that groundwater onto the local reef. This is a very natural process. It's essential for proper coastal ecosystem health and development. However, it can become problematic when this groundwater becomes contaminated with fertilizers, pesticides, pollutants, or even septic seepage in such a way that these contaminants can make their way out onto the reef. And this is essentially problematic for ecosystem and as well as human health, because this water can contain sewage bacteria or other fecal bacteria, and it can also cause harmful algae blooms to occur. For the purposes of this talk, we will primarily be focusing on the presence of these sewage contaminants on reef water and different types of reef microbial ecosystems.
How does development in the Pacific Islands impact coastal environments? Some of the community needs, both in Hawaii as a Pacific Island and in Tutuila, were to identify vulnerable areas to algal blooms and assess groundwater impacts on the reef, as reefs are one of the factors that we use for sustainability in our everyday life. Some of the tools that we used were actually developed at UH Manoa and in Hawaii. So we're going to be using hydrological tools, microbes, and lemurs. So why Tutuila? Tutuila offered a similar environment to Hawaii, but it had four different locations. So we had Pula Lagoon, which was our most developed. It was high density and it was a mix of septic tanks and sewage lines. Funga'alu was not as densely populated as Pula Lagoon, and it did have a sewage treatment center. Vatia was a country village. It wasn't too populated and mostly contained in itself and in the coastal area. And then O'a was our pristine site. That site was uninhabited, just a few bats on some of the nearshore trees. So we deployed starved algae. What that means is that we took algae that was natural in the environment and we depleted them of their nitrogen. In the next few slides, Becca will talk about what that actually means. We also took 2 different coral samples. We deployed bricks with slides on them to see what kind of biofilms would grow. We also used a YSI machine to measure things such as salinity of the water, pH, and temperature. And we also took bottle water grabs. What that means is just we stuck our hands into the water about 30 centimeters or so, and then just capped the water and then took it back to the lab to process.
So one of the things that we use these measurements for is to measure nitrogen, which is an essential nutrient to coastal waters. One of these measurements, delta N15, is a ratio of heavy nitrogen to light nitrogen. And we use this because our bodies and the bodies of animals preferentially incorporate lighter nitrogen, and we excrete the heavier forms of nitrogen. And so we can use the delta N15 values, this ratio, as an indicator of nitrogen source. Anything at or above a value of 9 can be considered as coming from a fecal source, so manure, a septic tank, or a water treatment plant. From our LIMU deployments, we see that we did detect some delta N15 values above 9 in Pulla, and somewhat unexpected in Vatia as well. From the water samples, so these are more transient, there's a lot more flow in and out of the area. We also detected delta N15 values at Pulla, and as well as one site in Faga'alu. Another nitrogen measurement that we used was the nitrogen percentage. And this indicates the amount of nitrogen going into and out of an area. So a greater nitrogen percentage, there's more nitrogen flux. As expected, Pella and Faga'alu had a greater nitrogen flux into and out of the area than Vatia and Oa, our pristine site. Another measurement we took was the carbon to nitrogen values. This ratio represents the cost of growth. So a high carbon to nitrogen ratio is indicative of more restricted growth. It's more expensive to grow, versus a low carbon to nitrogen value means there's unrestricted growth. It's very cheap to grow. And so populations will fluctuate more rapidly. And this is where you can get the large booms and busts associated with algae blooms. And then, as we expected, we saw that Pella and Faga'alu, our more developed sites, have this lower carbon to nitrogen ratio associated with unrestricted growth as compared to our less developed and our pristine site. So in addition to these environmental nutrient samples, we also collected microbial assemblages. You might know that coral has a unicellular algae, but you might not know that coral also has a plethora of bacteria that lives in its tissues that is absolutely essential to coral health, just like our gut bacteria are essential to us. We sampled 2 coral species, Parietes cylindrica and Passalopera demicornis. We also sampled the biofilms growing on the horizontal and vertical glass slides, as well as the bacterioplankton communities, just swimming freely about the water. We took these samples, we isolated and sequenced the DNA, and we used a variety of different methods to turn all of these millions of DNA reads into something a little bit more understandable. One of the things that we do to compare samples, for example, is to generate trees. So this is kind of like your family tree. So in this mock tree, sample A and sample B are more similar to one another than sample C, but altogether, the 3 are more similar to one another than either of them is to sample D or sample E. Another thing that we'll do, in addition to these trees, is to add a heat map, which is a visual representation of different microbial groups here on the y-axis. For example, we have groups 1 through 5, and it applies a color gradient that indicates presence or lack thereof. So for example, sample A here, this column, we can see that sample A is predominant in microbial group 1, microbial group 2, and microbial group 3, but less so for microbial group 4 and microbial group 5. And that by looking at these heat maps across all of the samples and different microbial groups, we can begin to pick out patterns. So here are the trees that we were able to develop for the water and biofilm samples. The leaves here are color-coded by the watershed in which they originated. And one of the immediate things that you might notice is that a lot of the sites group together. So for example, with the exception of only one case, all of the Pulla sites group together in sort of these watershed clusters. And that the clusters in the biofilm are a lot cleaner than they are in the water samples for all of the other sites. Adding to this then the heat map of different bacterial phyla here on the y-axis, you might begin to see that there are some microbial groups that are present across all watersheds and all sample types. And then there are others that are present predominantly in the water, not so much in the biofilm, or vice versa, present in the biofilm, not so much in the water. You might also begin to see where, looking down, you might pick out some of these particular boxes where you can identify microbial groups that are more present in polycytes or in Faga alu, Vitia, and oocytes, simply by looking at these larger scale patterns. And then for the remainder of the talk, we are going to be looking at 4 specific groups located here on the heat map. These groups are Enterococcus, Streptococcus, Staphylococcus, and Leptospira. Now, before we proceed any further, I would just like to have a few disclaimers. One, the types of analysis that we run are all relative abundance. These aren't meant to be mistaken for absolute counts. And second, these are just the presence of these groups. Our analysis cannot confirm pathogenicity or confirm the presence of fecal material in any of these samples. So results are to be taken with a grain of salt.
So we detected fecal indicator bacteria in all sample types. The 2 fecal indicator bacteria that we kind of focused on were Enterococcus and Streptococcus. The reason is that these are well-known and well-established bacteria when looking at fecal indicators. So for our particular results, we found both in water films and water. And what we saw was that Pala Lagoon, kind of as we hypothesized, would be higher, followed by Funga Alu, Vatia, and Oa. What was interesting was that we found across several sites and in kind of a higher abundance at Vatia, making this kind of an interesting find for Vatia. We also detected Leptospirea. Most people in Hawaii are familiar in that Leptospirea is linked to the opportunistic pathogen leptospirosis or Wells disease. Hawaii actually accounts for 50% of the cases reported in the US. For our particular Tutuila samples, we found Leptospirea in biofilms. And what was kind of interesting was that we didn't find too many in Funga Alu. We found some in Pala, but a lot of our samples in several kind of like a higher abundance came from Vatia. So that was interesting as well. We also detected Staphylococcus. Again, if you're frequent in the water in around Hawaii, you are familiar with Staphylococcus, and it's linked to the opportunistic pathogen staph infections, which is mostly a topical skin disease, sometimes causing upper respiratory tract infections. For our particular data, again, we found that Pala Lagoon, as we hypothesized, did have a lot of Staphylococcus in the water, and it was at higher abundance. We didn't see too many at Funga Alu. Interestingly, again, we saw a lot more than expected in more sites in Vatia, and then again, as we hypothesized, pretty much none in Oa, as it was our pristine site.
All right, we have a lot more work to do, but we hope one of the key findings that you take away from this talk is that contaminated groundwater is a major source of nutrients and pollutants to coastal waters, as well as a contributing source to microbes to coastal waters. And that trends in nutrient loading did follow our expected patterns based on development level for each watershed. Biofilm and water microbes do exhibit different patterns. So it is important to keep this in mind when planning future experiments, but they do generally all group by development level in these watersheds. We also found higher than expected nitrogen levels and microbial groups containing fecal indicators and opportunistic pathogens and Vitia, which does indicate a potential hotspot for future anthropogenic change, and definitely a site we want to look at more in the future.
So for future directions, we kind of wanted to take our data and focus. We wanted to hone in on the potential hotspot of Atia. We did do some algal deployments, more biofilm deployments, which was the slides, and then more water sample grabs. We're currently in the process of analyzing the data set, and hopefully we'll be able to present it to you sometime soon.
All right, with that, we just want to say a big mahalo to our local on-island support team, particularly the villages of Afono, Vatia, and Faga'alu, the American Samoa Community College, National Marine Sanctuaries, our absolutely fantastic field team, as well as our funding source, the Water Resources Research Center.
For logistical support, we would like to thank all the people mentioned. We'd also like to thank the Center for Microbial Analysis through Island Knowledge and Investigation, CMIKE, the Hawaii Institute of Marine Biology Genetics Core Facility, and then a big lava to the East-West Center, particularly the International Graduate Student Conference organizers. Thank you for having us and allotting us a time.
Apologies, I think we're saving questions till the end, so I'll just stop it there.
Thank you. Thank you, Becca and Ashley, for your presentations. So now we still save the question to the end, and then now we move on to the fourth one. So the last presentations of today. Waka Armas, are you here? Waka. I don't know if he's here or not.
Let me message him on the chat because he's in the room and has been messaging me, but he's been having issues.
Yeah, I haven't heard from him since beginning, so
I'll shoot him a quick message, and then if it doesn't work, I have a copy of his slides that I can share and a recording.
Oh, yeah, please. So we are going to have Waka Ama with his presentations on role of institutional environment in developing effective learning organization culture, an empirical study of higher education institutions of Pakistan. So Waka Ahmad is from Management and Economics Departments of Kunming University of Science and Technology, China. Not sure if he's here or not.
Okay, so I'm going to share from my screen. Can you see okay?
Right, yeah.
Yes, okay, great. I'm gonna go ahead and play it then. So this is Walker's presentation.
Okay,
Hi,
thank
everyone.
you. Oh.
Hopefully you all will be fine. I'm from Wakarimut. The topic of my presentation is role of institutional environment in developing effective learning organization culture and empirical study of higher education institution of Pakistan. The background of the topic of my research is that the organizations are passing increasing pressure from the institutional environment to the aspect of a learning organization within their arsenal. So various social and cultural pressures have impact on the organization to adopt different strategies that are necessary for an organization to become a learning organization. The previous research considerably discussed institutional environment and its impact on various sectors of organization like business, CSR, HRM. But little attention is paid to the relationship of institutional environment with learning organization culture. Based on the argue that the majority of the studies in the field of corporate social responsibility only focus on the section between institutional environment, assisted learning, e-learning, or CSR financial performance, while ignoring other factors of institutional environment and learning organization. So it means that this is the new relationship. On the second slide, that's the part of study background and introduction. This is a unique quest to probe how an institutional environment help influencing institutional higher education institution in developing an effective learning organization culture. According to the research report, only two Pakistani universities out of 188 made it to 500 universities, while merely seven made it place in the top 1,000 university. This indicates there is a lack of quality research, quality faculty, lack of quality faculty members, lack of training capacity, education corruption, political involvement, and nepotism, and overall lack of institutional structure which couldn't lead the higher education institution in developing an effective learning organization culture and higher education at the higher education institution. So there is a need to address all institutional environment factors in the developing a learning organization culture in the context of Pakistan. Uh, Based on the previous problems, uh, I'm going to explain what is the basic problem of higher education institution, why I select this topic for my research. Similar to the contemporary business organization, higher education institutions are facing immediate pressure to survive. Unlike business organization, the core activities of higher education are research and teaching. However, what sets a top university apart from its competitor is the quality of performance concerning knowledge production in the form of building the learning organization culture. However, higher education institutions are continue to operate on fiscal issues like transparency, quality, number of graduates, lack of quality learning, and unsatisfactory performance for the students. Furthermore, in the context of Pakistan and exodus of students from foreign countries is also staggering. Research problem in unemployment are main problem due to the lack of learning organization culture. Such objective. The study attempt to develop a model to analyze organization culture based on the institutional environment, cohesive, normative, mimetic in higher education institution of Pakistan. To study the institutional environment that will enhance the learning organization cultures and concept in higher education institution. Analyze the dimensions of learning organization culture and its antecedents to identify a diverse competitive outcome for the higher education institution through the institutional environment to become having a learning organization culture, to know about the implication and potential challenges for the higher education institution to become a learning organization with respect to institutional environments. These are the research questions related to this study. What extent an institutional environment affect learning organization culture in a higher education institution of Pakistan? How institutional environment factor affect on learning organization culture? What institutional which institutional environment has the strong effect in promoting learning organization culture in higher education institution? These are the hypotheses. Four hypotheses are the part of this research. First one is there is institutional environment of an higher education institution is an important factor that affect the underlying approach to learning organization culture. The second hypothesis, an organization who has higher cohesive pressure are more likely to behave promoting like a learning organization culture. Third one is that organization who perceive higher normative pressure are more likely to become having a learning organization culture. The fourth one is higher education institution who perceive higher mimetic pressure are more likely to become having a learning organization culture. Significance of study. The first one is that a systematic review explained that this is the first study to discuss the relationship between institutional environment and learning organization culture. Secondly, the Pakistani government and governing bodies for higher education institutions are now paying increased attention to the learning organization concept, which emphasizes building institutions with better performance through a learning process and research etc. However, an extensive literature review has identified there is no such study on institutional environmental learning and its culture for Pakistan higher education institution sector so far. So this is the new one. Thirdly, this study also play a significant role in enhancing education level of higher education and also guide the main stakeholder think about the education sector problems seriously. Lastly, this study employs instrument that has not been previously tested in the higher education institution of Pakistan. The treasure review. The institutional theory is firstly introduced by Liu Yimako and Powell in 1983. Institutional theory study organization forms that explain why these forms are isomorphic within an organization field. Organization field is defined as a set of organizations that in the aggregate constitute a recognized area of the institutional life, key supplier, resources, product, consumer, regulatory agencies, and other organizations that produce similar sources or products. Within an organization field, organizations are considered as conforming due to the increased pressure for change. By doing so, organization will reap the benefits of resources, legitimacy, and survival capacity. Slides. We can say that institutional environments are characterized by the elaboration of rules and requirements to which an individual organism must conform in order to receive legitimacy and support. Coercive pressure or formal and informal pressure exerted on the organization by other organizations upon which they are dependent and by culture, expectation in the society within which organization function. Normative pressures arise from the professionalizations uh, defined just like Bimova and Lawson defined that normative pressure as a force that comes from the common intellectual base of shared understanding, definitions of the norms, and legitimate actions within the professional arena. Mimetic pressure results in the organization for to copy or imitate the other firms or performing best in the market, or in other words, to follow the bench. Learning learning organization applies where people continually expand their capacity to create results they truly desire, where new and expansive patterns of thinking are nurtured, and where collective aspiration is free, set free, and where people are continually learning how to learn together. Learning organization culture serves as a sense-making mechanism that guides and shapes values, behavior, and attitudes of the employees. Research methodologies. For the data collection process, a mixed method approach was used. Target preparation was higher education institution. A Pakistan survey and review approach were used to collect the data from the respondents in higher education. Faculty members, director, deans, policymakers, and postgraduate students from various higher education institutions was used as the accurate respondent in this study. Purposes sampling was used to collect the data from the 245 response respondents in higher education institution of Pakistan. In the interview part, 15 interviews were conducted with the main stakeholder like university chairman, dean, quality enhancement cell director. Both descriptive and inferential statistics were used to analyze the data. Content analysis was used to analyze the interview data. Institution environment scale was adapted from Chu and Cheng 2013, Fan and Bird 2015, Majid et al. 2019. Learning and adjusting are adapted from Cinch 1990. This is the study purpose model. Institution environment. The sub-dimensions of institution environment, cohesive, normative, and mimetic, and learning organization is the dependent way. Result of the demographic information is that 70% male and 30% female gave their responses for this study. Further, uh, 73% graduate, 27% postgraduate faculty member recorded their responses. 45% employees have experienced more than 5 years. 30% faculty members have experienced more than 10 years. 25% faculty members have the experience more than 20 years. Most of the young faculty members gave their responses. Interviews were conducted with 7 professors, 3 department chairmen, 3 directors, 2 quality enhancement heads. This table shows that there is a strong and positive relationship between normative pressure, mimetic pressure, coercive pressure, and learning organization culture. The second one is this is the regression table. Table shows that independent variable like normative pressure, normative environment, mimetic environment, and cohesive environment explains a variance of 67% in learning organization. In this table, we can check on the value of beta and value of rho and t statistic. All the independent variables has a positive two mimetic environment and coercive environment has a positive and significant relationship with learning organization, but normative pressure have a negative but significant relationship with learning organization. The finding, the purpose of this research was to test the model of institutional environment and learning organization culture in the higher education institution of Pakistan. Results showed that all the independent variables have a positive and significant relationship with dependent variables. The study results show that all the institutional environments, normative, mimetic, and cohesive, bring a change of 67% independent variable learning organization culture. Study results explain that all the research hypotheses that have accepted, supported by this study. Most of the interviews have the views that the institutional environment has a positive impact in promoting the learning organization culture in higher education institution of Pakistan. Study result explained that the institutional environment had a positive and significant impact on learning organization culture. Further study findings add a new theoretical contribution to the literature of learning organization culture with respect to higher education institution. What are the implications and future research directions? The study ultimately have implication for the practitioner like higher education institutions and regulators and researcher to further research and practice the concept of learning organization culture in higher education institutions and other sectors as well. The study gives a new direction for the other sectors to research on these issues with this theoretical model. The study gives a new theoretical contribution in the theories of learning organization and institution. It will be a new addition in these two theories body of knowledge. The findings of these studies are expected to be applicable for similar developing country context where organizations generally do not face many pressure to develop and learn, especially when it comes to higher education institutions. Conclusion, the current study found a direct relationship between institutional environment and learning organization culture. There is a positive and significant relationship between institutional environment and learning culture, and the study provided significant addition in the literature of institutional theory and learning culture. The result of the study can be a strong policy application for the higher education institution, department, authority, managers, supervisor, and other related parties by providing practical guidelines to design tasks associated with institutional environment and learning culture in the context of higher education institution of Pakistan. Thank you for your patience. If you have any queries, any questions, you can consult me. Thank you. This is
Thank you, Waka. So now, thank you for all the great presentation. So now we move on to the Q&A sessions. Do anyone have questions? Yeah, I think there one question for Serena. Do you see in the chat box from Natalie? Yeah.
I do.
Yeah. So the
Should
question.
I read it? Oh.
Yeah, I can read the question for you. Yeah. So, to the best of your knowledge, are there any initiatives to use the data found in the TPN in schools at the elementary, middle, or high school levels? If so, how? And if not, then why do you think that this is the case? Yeah, so go ahead and answer your question.
Yeah, thank you, Natalie. This is a fun question because it can allow me two kind of different angles in. The first one is, to my knowledge, I don't know any use of the data in schools. I'm sure there is, but I think one of the challenges that the TBN faces is that it doesn't have a great job of showcasing the research that it's used for. Like, it shows how many times data sets have been downloaded off the site, but there's no real connection between who gathers the data, who uses the data, what it's used for. So I think that's an opportunity, the lack thereof of me being able to tell you an answer to that question. And then, like, why do I not think that is the case? Or I don't know, I guess the reason I can't give you an answer, I think that explains that well enough. Did I get your, like, the first part of your question, Natalie? Yeah. And then the other thing is, whereas there's not, any clear way to know whether it's been used in schools, for sure there are school groups going out and doing citizen science and contributing to the site. So I think, especially even just in the community that I know well, they actually integrate their community science initiatives with elementary and high schools, especially, yeah, elementary and like junior high, actually, less high school. Yeah, they introduce these kids to their local environments by way of citizen science, and also it gives them the chance to tell other stories, which I think is really the intent behind a lot of these organizers. They want to get these kids out there to learn about local history, and the interconnections between life is kind of a byproduct. Yeah. Thank you.
Thanks, Serena. Do anyone else have your questions? You can just unmute yourself and ask the questions. Or you can write in the chat box.
Yeah, I have a question.
Yeah, go ahead.
So, my name is Kanokon Latanapon, and you can call me Bao. So my background is in social science and political science. So my questions here will be related to how this is to all of the presenter, how your studies are related to the local communities or the study group that you're related to. I would like to hear about your impressions about the community that you work with during your studies, and are there any challenges during your study and during your with the communities, and I know that all of your studies are really precious. That your fighting will help the community, especially for the one in for Kimberly, right? That then you find a new species and then you help them for the aquaculture. So how do you raise the awareness, the community awareness about your the benefits of your studies, and eventually I think it will help them to realize that they have the potential, right, to help, especially like what Serena talked about, citizen side. I never know before that when like every people like me can become like a scientist, and we do not know that our thinking process can fit in that scientific study. So yeah, I would like to know just this like basically 3 questions. What are your impression about working with the community? What are the challenges? And then how do you raise the awareness of the communities about the benefits of your studies that will impact the community immediately? Thank you.
should I go first? Or yeah, okay. Really good questions. Thank you. So yeah, so I did face, and a lot of researchers in Fiji face a similar challenge. So one of the main ones that I faced is along the Navua River, there are different villages, and gaining access to that section of the river where the village is located can be a bit tedious. There's a lot of traditional protocol that needs to go into place, and whether they allow you to use it or not is another thing on its own. So that proved to be quite time costing. So my section of the study was within an area that wasn't dependent on village access. Apart from that, you had asked how the study would benefit them. I think another challenge is locals. They still don't understand that traditional fisheries is that fish stocks are depleting, like wild fish stocks, right? And they don't understand the need for aquaculture because all their life they've known, we'll get on a boat, go and catch our fish, come back home. Oh, and so yeah, trying to get them to understand the importance of aquaculture, especially today, is another challenge. So after the research was conducted, our next efforts, we've been in contact with a few local villages just outside of the city center, so in the rural areas, and we're trying to culture reve in their areas with the least cost to them as well, right? Because that's important too, finances. So that's the next phase of this study, and I think it will help in the long run. So I hope that answers your question. Sorry.
Just because it's like, so can I just pin on a question for you, Kimberly, on the same exact thread that Pau started on? I just wanted to ask, where do you think these aquaculture sites would end up, and like who would own them, I guess, was the question I was thinking of.
All right, so ideally, like our goal is for villages to own these aquaculture sites, because so the issue here is the only aquacultured fish are mainly tilapia, which is very common globally, right? But it's, for us anyway, we're used to very salty tasting fish, right? So tilapia is a bit bland to our palates. So we're trying to find a fish species that want to culture because they like eating it, right? And we know that reve is already found in markets, locals already eat it. So we're trying to find something that they'd be happy to culture. So yeah, the main goal would be for local villages, especially to aquaculture this species, to replace or to sort of put pressure off wild stocks, especially. Yeah.
Thank you so much, man. That's so cool to hear about. Thank you. I'll try and answer the question just because I already got myself in here. But yeah, I would say I had a whole year of getting an impression of the community, not because I was doing research, just because I lived there. I was living in southern Taiwan, a place called Kaohsiung Meinong. And man, my impression, if you read the article that I linked in the chat, or that BU helped me link in the chat, you'll see that this community really doesn't need me. I'm not like doing something like Kimberly that will like genuinely help how things work on a day-to-day basis. I'm more trying to amplify what they're already doing to help themselves, because they're doing restorative ecological projects in the forest nearby, but also what I write about in the article is about kind of bird like a bird habitat that was restored to allow a locally absent species to return to the town. So yeah, I think the impression I have is very positive. They're able to solve a lot of their own problems on their own. A challenge identified by the person I interviewed was that young people are not as engaged in these issues as older people, especially farmers who are kind of on it and thinking about the future. And then I think, yeah, the challenge that I feel like my research actually faces, which is not not about them getting their needs met, is more like, how do we just let other people know the existence of Taiwan and like the power of environmental movements there? Because it's not really like, we don't talk about Taiwan nearly enough, in my opinion. And I think that there's a lot to learn in terms of digital democracy, deliberative democracy. Yeah, and I think looking at it through an environmental and ecological lens is also really powerful and informative.
Thank you.
Any other questions? Nalini, go ahead.
Oh, okay, I think I can speak for the American Samoa. It's similar to Kimberly, where we have to go into villages, but American Samoa is a little different. It's governed, but then they still have villages that have their own sense of government. I was lucky because my family is from American Samoa, so I knew that we had to reach out to like pulinu'us and do certain protocols. And it's really based on the grace of the village, the matai, and the pulinu'u, whether we come in, and it's a day-by-day thing, and we have to be sensitive to that. As for our question, it actually came from both the villages as well as the government. So we were lucky that it kind of, water quality was an issue that they wanted to look at, and their reefs they wanted to look at. So that was totally community based. And then our information goes straight to both the government as well as the villages in an appropriate sense. Yeah, I think, was that everything?
Hi, I have a question for Becca. Thank you for your presentation. It's very impressive. I just want to ask, I noticed that you used the horizontal and the vertical plate to collect the biofilm. I just want to know, the biofilm you collected, are they different for the microbial? Are they different or are they the same? Thank you.
Yeah, thanks. We actually have run some statistical tests and we have confirmed that they are correlated, the horizontal and the vertical, but we do see different patterns in both. And we attribute it largely probably to the horizontal biofilm. In addition to what's growing, things will settle on top of it, versus things on the vertical biofilm are definitely just growing. But yeah, it's very interesting, and it might be due to smaller things like light attenuation. For example, for the presence of cyanobacteria, which use light for energy. So maybe they're more productive on the horizontal biofilms versus if they're like sideways on the vertical. So yeah, they are different. That's why we wanted to do these two different methods types as well, because we wanted to ask questions about, are these comparable or are they different? And is this something that we need to take into consideration for future biofilm studies? Yeah, thank you for your question.
Thank you very much.
Hi, I have two questions actually for Becca and Serena. So maybe I'll start with Becca. So in your studies, were you only considering the populations of the area, or were you also factoring in the advancement in development like the sewerage system? And you conducted 3 studies. Did you consider any control experiment, like something, a reference point, or was it not needed?
Yeah, thank you for your question. And Nalini, you can jump in if you have any additional things you want to add on to this. But doing environmental monitoring is a little bit tricky because we don't have a control in the sense that we would if we were running an experiment in the lab. However, for the purposes of our study, we do have sites around the Hawaiian Islands. That's how we developed these tools. So in a sense, we kind of knew what we were looking for, but for the purposes of the islands on the island of Tutuila, we considered OA, which had absolutely no human development, as sort of our control site. We call this the environmental end member, just to compare it to any of the previous development levels. And we used a variety of different factors to quantify development. For example, we looked at the number of buildings and their distance to the shore for each of the watersheds. We did not show that data, but we do have that when we run more of like mathematical models.
Okay, thank you. My next question is to Serena. Your study was very interesting, and I was wondering if you were to scale this to different countries, maybe not specifically Taiwan, but to different geographical locations, what do you expect will be your greatest challenge, or your greatest challenge? What would that be? If you were to go to different communities, set up geographical locations.
Can I clarify the question? So if I were to do kind of more kind of investigative research on citizen science in two different communities outside of Taiwan, what do you think I would?
Yes.
Oh, well, I mean, citizen science, I think, is done differently everywhere. And I think the Fan and Chen, they're like really useful terms of CS1 through 3 that I showed you, kind of gives an idea of why people are doing it, and that sort of shapes what they do. So I, for example, would fall into the CS1 category. I do the data collection I do because I'm like, oh my God, climate change. And so I go out on a walk and I take pictures of everything I see and I upload it and I identify it and I work with other people to make that a fully open data point. So that's maybe how I do citizen science. And so I would like know that when I see it in a community, if that's what's motivating people. And I think if I were to go somewhere else and like the real thing you need to do is just ask like what people's motivations are. Grace actually asked a really great question in the chat sort of about this, of like things that you could ask to get an idea of what's motivating people and sort of how they situate themselves in what citizen science is. But I think it's hard to answer that question because I can't visualize two different places that I would really go. And my current work, as you saw, was much more like, it was kind of a structured lit review. So I wasn't in this study like going into the community necessarily. The initial thing that I presented last year was more of that. But I would say like, if I were to go in, the challenges I would face would be like linguistic probably. And, you know, making sure that people can find some connection with me in terms of like how this will be useful in like making sure that their needs as a community are met, like the things that any, I think, researcher would have going into a community. I don't think my topic makes it harder or easier, really. Did that kind of get to your question?
Yes, thank you.
Also, I said this in the chat, but oh my gosh, Becca and Nalani, your presentation, just the visuals, the logical flow, just, man, you made me wish I had recorded mine as well.
Thank you.
Yeah, thank you, everyone. Do anyone still have your questions?
Hi, sorry, I just had one question.
Yeah, go ahead.
For the microbe indicator study. I don't know if you covered it or I don't know if it's in the range, but I was just curious, especially for the infected microbes, is that what it is? Like how does that affect, does that affect the health of communities in the areas that you studied? Because I saw that you had like staphylococcus and those kind of things, which I know is like quite a common bacteria, is it? So yeah, I just wanted to know if that had an effect on the health of communities in the area that it was present.
Melanie, you want to take this one?
No, you can.
We don't have numbers for the, like, for example, the amount of staph infections in the villages or anything quantitative like that. In fact, for a lot of the more remote villages, I don't actually know if that's something that they keep record of or have medical records for, as sometimes these villages would have sort of like a, you know, self-treatment for some of these infections. But we do know that there are a lot of social disparities between the amount of, like, for example, staphylococcus infections. They predominantly, or at least significantly, infect more Native Islanders and homeless populations than they do in some of the more urban areas. But yeah, that would be really interesting if we could somehow figure out a way to have those numbers.
I do know that yeah, so like the remote villages, like Vakia itself, they won't go to hospitals. There's only one hospital on Tutuila, and a lot of people don't have insurance, so they go to somebody who does full ma'i, which is like a medicine person. And then I do know when we were there, they told me so the first year the first team went, they didn't tell them anything, but when we went to Vakia, because I was speaking with them, they told me not to drink the water and to boil it. So I think there's some self-awareness there, and then they're just not advertising it. But yeah, so that's our only kind of measurement we had.
Yeah, it would be really interesting to do this study in more Pacific Island countries, because our water pretty much everywhere is not of great quality, and our health services aren't great. So it would be good to see the relationship between how it actually affects the health of. Thank you, you guys. Presentation was really good. Thanks.
Yeah, and last chance for questions if anyone still have.
Can I have a
Yeah.
I think I
Sure.
have a questions for Waka. Are you still there? Yes. So
I'm not sure if he's still here.
He is still here. He cannot hear us any longer, so he asked me to basically type the questions to him, and he will try to respond over the chat. So we'll try.
Okay, so I typed the questions in the chat here or on the whole
yeah.
blog? Just type it in here, right?
Sure. Yeah.
Yeah, I think in the Zoom.
Okay. All right. Thank you.
Yeah, meanwhile, I just want to remind everyone that before we end the session, please go to the form, the feedback form that Megan just sent us in the chat box, and fill out the feedback form before we leave the meeting. And now we're back to the question for Walker. Did you send him directly, Paul?
It muted you. Say it again.
I'm typing. Sorry.
Okay. Yeah.
I put it in the
no
chat.
problem. Yeah.
Yes.
Does anyone have particular presentations coming up in the next few sessions that they want to share about that we should go to?
I don't. but I will be on almost every session today.
I believe that on the Huba platform that we that you should have used to sign up for this and everything, I think there's a way to connect with other participants. So Pao, if you're not able to get a response from Walker here and now during this meeting, you might be able to reach out to him on that platform to get some more conversation.
Okay. Okay, I think I posted to everyone. It's just a short, pretty short questions. I think he has already reviewed this.
Yeah.
I think, yeah, just from his subject of the study, I'd like to know then what are really the most challenges.
Can you connect it? Megan, can you talk to him or not? Because he's still muted.
Sure, yeah, so I'm just getting direct messages from him. So I think there's a bit of a lag as well. It seems like the internet connection isn't great. So yeah, I'm not sure if we'll get a response in time.
Yeah, we can wait for him for like 1 minute. If he did not respond, we will just move on. While we're waiting, so I just wanna remind that we will have the IGSC closing ceremony and closing panel tomorrow at 3:30 p.m. So please join us if you can. We have something in the chat box. Oh, so Waka sending the answer by chat. From Waka, actually in our country, we have different problems like lack of funds, lack of quality research, awareness, and motivation. So kind of like lots of challenge from that side. And Kenvin Rickman. Wow. So continue the answer from Walker. No such type of training and coaching which can help in promoting learning organization culture. It's kind of like challenging field. Kenvin Rickman, do you wanna unmute us and ask your question?
Sure. I was just curious about how we can go from the scientific perspective, relaying our results of studies and whatnot in the scholarly format. How can we change the way we discuss these things and frame the results in such a way that people who are affected by these issues, biodiversity loss or lack of arable land for farming, how can people take these studies and translate it to power in their local policy decisions, government, etc.?
Thank you, Kevin. This question is for everyone, right?
Yes, in general.
Yeah. So our presenters, anyone want to answer this question?
It's such a good question. That's why it's so hard. We're all just like, how does one begin? In terms of the framing, I'll just like give it a go and give you guys more time.
Yeah, feel free.
Yeah, I would say, I think our academic voices have potential to amplify things. It's hard to say how an American in California can influence the strength of a voice of someone in Taiwan or in really anywhere. Like what power does that have? I think like that's the first open question. It depends on kind of the policy environments in both places. I think in Taiwan, there's a lot of fear, anxiety about how Taiwan is received on the international stage. So I think anything that would make the Taiwanese government look bad, they will do something about. Not the case everywhere, I will say. I can only speak sort of to my experience on that. But yeah, I think like in terms of framing, just making sure that you're using quotes from those people directly when you're doing interviews. We talked a lot about like the gift giving thing. I think like asking, sometimes for me, the best gift is one where you ask what someone else needs and then give that to them. So I'm working with the person I interviewed on a piece to explain, basically an explainer in English about the restoration project they're currently doing. And yeah, just kind of like taking the direction of what they want and running with it, I guess. But that's all I can think of in this moment. Thanks for the question. Definitely one to meditate on much longer.
Thank you.
And if you have ideas, hey.
I have a few, but we can talk more outside.
And to kind of jump onto what Serena said, yeah, definitely starting with what the community needs is incredibly advantageous, because then to a certain respect, you're already speaking the same language, and that's the language of what does the community need. So in our case, we were extremely fortunate that rather than how it usually works in science, it was the community that approached sort of the scientific community and hydrologists that were currently working on the site to start asking some of these questions. And then it's also additionally advantageous, and at least on Tutuila for us, because once we inform the villages, they maintain a great deal of power in self-governance. So if there is an immediate problem, they have the power to more directly change it. There's a little bit less governmental red tape, and it's more of a just knowing who is important to talk to in a community for them to be able to make changes for themselves.
I do want to note that sort of explaining our science to the community was a little challenging, even with native like Samoan speakers. A sense of making the community aware of our findings, and then I guess it took a while for the community to kind of observe it, and then there's like a sense of pride in the village and just letting them know that it's not something to be ashamed of, I guess, because they feel like they're caretakers. So really just straddling that line and being super sensitive, and then letting them tell us what direction to go, and then we use our contacts to kind of help where we can.
Excellent. Thank you. Great answers.
Thank you everyone for great presentations and your active discussion today. I learned a lot from this presentation today. Thank you. Have a great day, and don't forget to fill the feedback form with the link in chat box. I just copied the link and paste back here. Yeah. Thank you. Have a good day, and maybe see you in other sessions. Bye, everyone.
Bye, everyone. Thank you.
See you. Bye.